Hi, welcome back again, and this is the second part of our discussion on membrane structure and function, membrane transport, and cell junctions. So last time I introduced the idea of selective permeability, which, remember, is that some substances can move freely across the membrane, others cannot move freely, and that means they have to be helped. I also introduced the idea of a model of the plasma or the cell membranes called the fluid mosaic model. So I explained the mosaic part. Now what I'm going to do is help you understand how membranes are fluid, and not really fluid like the way the water's fluid. They're said to be semi-fluid. In fact, they have about the viscosity of olive oil depending on temperature. So what makes membranes fluid? It's to do with the motion of the phospholipids in the bilayer. These are not static structures. There is a lot of kinetic energy. There's a lot of movement of the phospholipids. So phospholipids can move three ways. They can do kind of side-to-side motion, so they can move around in the plane of the membrane completely freely. Now, sometimes they're going to be restricted to certain regions of the membrane. Different membrane phospholipids, remember, can have different types of polar head group, and so you get these little kind of rafts of different types of lipids, and you get into that in cell biology, so we're not going to get into that. But the membranes can diffuse laterally, so they can move this way. If this is the plane of the membrane, then they can move like this. If these are the heads, they can move like this, and obviously that amount of motion is going to be directly proportional to heat, because that is going to give us kinetic energy and movement. The other thing that phospholipids can do is they can spin, so they can rotate around an axis. So if my hand here is the, well actually let's say this, here's the phospholipid head, and imagine my arm and my wrist are two fatty acyl tails. They can do this in the membrane and move around kind of like this, diffusing everywhere. So this is diffusion. This is random movement due to kinetic energy. Here comes Eddie. He's obviously interested in membrane lipids. There's also a third type of motion that you can see, but this is very rare, and it has to be helped by a special enzyme. And I'm not making this up. The enzyme is called a flipase. And what a flipase does is it uses the energy that comes from the breakdown of ATP to take a phospholipid, which has got its polar head group on one side of the membrane, and then it flips it through the membrane to the other side, to the other leaflet. So you've got to bear in mind is what we're doing there is moving a polar head group all the way through the hydrophobic core of the membrane. Now that is thermodynamically very unfavorable, and so what we have to do is put in some energy to do some work. So those are three types of motion: lateral diffusion, rotational motion, and then with a much lower probability, this thing called flip-flopping, where the membrane lipid goes from one side of the membrane and it just flips, or flops if you like, to the other side. So the next thing to talk about really here is what affects membrane fluidity, because membrane fluidity changes based on lipid composition and cholesterol, and then some things about the phospholipid tails. So let's start off with cholesterol. But before we get into cholesterol, let's just go back to thinking about this molecule and its chemical properties, and therefore how it would find its way into a membrane. So here's cholesterol, and I want you to have a go at this question, taking a look at cholesterol, which I've left down in the bottom left-hand corner for you. What's the primary chemical characteristic of cholesterol? Okay, so let's get on to looking at factors that affect membrane fluidity, and we'll come back to cholesterol at the end because it's a bit of a weird one. Okay, so when you think about the fatty acid tails in a phospholipid in a membrane, the actual length of the hydrocarbon tail can vary quite significantly. It can be pretty short, or it can be a number of carbons longer. And so the length of that fatty acyl chain affects membrane fluidity. So why would that be? Well, think about it like this. If you've got all these really, really long fatty acid tails, those hydrophobic fatty acid tails, they're going to stick together, remember? Oily things like to stick together. So the longer they are, you can think about it as they have more kind of atomic surface area to stick together. And so the longer they are, the more they stick together. And the more they stick together, the less able they are to kind of have enough kinetic energy to actually be moving around. So what happens is as the fatty acid chain gets longer, they stick together, and that pushes the membrane fluidity down. So in organisms which you may find in warmer environments that aren't able to regulate their body temperature, you're going to find longer fatty acid tails to reduce membrane fluidity, because membrane fluidity has to be regulated very carefully. So this is a homeostatic mechanism where you've got to keep kind of membrane fluidity just at the right point, and if it gets too fluid, that's really bad. The cell basically melts, and if it drops too low and the membrane becomes insufficiently fluid, again, it's going to seriously affect cell function. So long fatty acid chains stick together, and that leads to a reduction in mobility, and that is a reduction in fluidity. If you shorten the chains up, think about it as though they've got less atomic surface area to interact. That means they can move around more freely because they're not sticking together, and that's going to result in an increase in fluidity. The other thing about the fatty acid chains that can affect membrane fluidity is the extent of saturation. And we talked about saturation when we talked about lipids, when we talked about fatty acids, sorry. So remember, if you have an unsaturated fatty acid and a certain type of double bond, you're going to get a bend in the phospholipid. So on the left is what you're seeing. On the left here, these fatty acid tails here, they're all dead straight, so they're completely saturated, they're dead straight. That means they can pack tightly together, and so again, they can't move around as much, and so they're less mobile, and that means that fluidity drops. Now, the flip side of that is if you introduce some double bonds, you take some of the hydrogens out like we talked about when we talked about unsaturated fatty acids, you start getting these kinks, and those kinks kind of push other fatty acids away from the kinked fatty acid, and so they can't pack tightly together. They're kind of all jostling and kind of getting their elbows out there, and they can't all pack tightly together, and so that means that the fluidity of the membrane goes up. So in an organism which is living in very cold temperatures, you may see an increase in the desaturation of fatty acids, so you get increased membrane fluidity. This is a very dynamic process. An organism which is going in and out of different temperatures and spending a significant amount of time in those temperatures, you may see changes in the saturation state of the fatty acid tails in membranes. So remember, where are these coming from? Well, we talked about where you're going to get fatty acids and lipids from. They're going to come from the ER. So there's a whole lot of conversation between the inside of the cell and the outside of the cell. Remember, we talked about how the cytoplasm, one of its jobs, is to integrate information that comes from the outside and give the cell the appropriate response or the appropriate behavior. That could be something like changing the extent of the saturation in the fatty acids made or the lipids made in the ER, so that the cell membrane, when it receives those phospholipids, has the appropriate level of fluidity. Let's come back to cholesterol. Cholesterol's a little bit weird. I don't need to overly focus on it. Cholesterol can do two things to membranes. One thing it can do is it can cause the fatty acid tails to get stuck together. And obviously when they get stuck together, that's like making them longer. That's like making them completely saturated. And that reduces their mobility and fluidity goes down. So cholesterol, under some conditions, can cause the fatty acids to pack together and reduce fluidity. Now at other temperatures, under other conditions, is what it does is it actually kind of pushes away the fatty acid tails, because it gets in there between the fatty acid tails and it kind of pushes them apart, and that allows more molecular motion, more fluidity. So cholesterol kind of does two different things under different circumstances. Don't worry about it too much, just know that under different circumstances it can have different effects. So what we've talked about here is membrane fluidity. We've talked about the extent of the saturation of the fatty acid tails. We've talked about the length of the fatty acid tails, and we've talked about the effects of cholesterol, and hopefully I've explained to you what it means to have a semi-fluid phospholipid bilayer in your cell membranes. We'll leave it there and we'll pick up next time.